Second type of energy is potential energy. This is stored energy. This could be a ball at the top of a hill, or the electrons on the outer orbit of a molecule. Chemical energy, or the potential energy of molecules, is based on structure for the most part, although we will see in later chapters how it can also be from location. A cell is just a tiny chemical factory. Some chemical reactions assemble monomers into polymers. Some chemical reactions break sugars and other fuels to release energy. Cells are energy transformers, converting one type of energy into another. Exactly. If energy could not be converted, we could not exist. Living organisms are matter, just as non-living things are. Energy transformations take place in living systems. Living systems are open systems. That means they interact with their surroundings. They take in light, heat, food, etc. These are exchanges of energy and matter. Systems and surroundings change depending on what's under study. For one researcher, a single beaver may be the system and everything else the surroundings. For another researcher, the colony of beavers is the system. A third researcher may be studying the entire pond ecosystem where the beavers are. That is her system. Living organisms convert energy. Plants convert sunlight energy into sugar energy. Mitochondria convert sugar energy into ATP energy. Conversion of energy, not creation of it. Words matter, so make sure you don't accidentally say create when you mean convert. Our system is our Earth. Energy comes into our system from the surroundings, the sun. This energy moves through our system in a one-way flow through producers, consumers, decomposers, then out of our system as heat. Even though some energy is lost as heat or random molecular movement, it's not destroyed. Remember that energy flows one way. Life is an open system. We take energy into the system as sunlight. It passes through producers, consumers, and ultimately decomposers. Each time the energy is converted into another form, some of it is lost as heat. The heat is released from the system. For a human system, the heat is released by sweating and evaporative cooling. Heat is a disordered form of energy. The release of heat makes the universe more random. It is random molecular motion, after all. The amount of disorder in a system is entropy. So energy conversions reduce the order of the universe and increase its entropy by releasing heat. In this conversion, well-ordered, high-energy molecules such as glucose are broken down into lower energy molecules and heat. This increases entropy. The increase in order as cells are built, organelles are organized into cells and cells into tissues and so on, is only possible at the expense of ordered forms of energy taken from the surroundings and released as a disordered form of energy. Compare the two processes above. You put gasoline, nice, expensive gas, into your car to gain forward motion. Some of the energy from the gas is lost as heat. Your money. What percentage of your gas energy do you think is lost? Also, look at the process of cellular respiration. Your food energy, more money, is being converted to ATP for cellular functions. But again, some is lost as heat. What percentage would you guess here? The natural conversion system of cellular respiration is more efficient than the man-created form of combustion. Of the glucose that goes through cellular respiration, about 40% gets converted to a usable form, and about 60% is lost as heat. Of the gas you put in your car, it's closer to 25% being converted to motion and 75% lost as heat. In both cases, however, you start with complex molecules and end with smaller molecules, converted energy, and random molecular motion. So both processes increase entropy. Here's a classic example of what not taking notes will get you. If I have a question on your test asking you to compare, contrast, explain endergonic and exergonic reactions, and all you put down is energy in, energy out, you aren't getting credit for it. So endergonic means it takes energy. Energy is a reactant. The other reactants are low in potential energy. You add energy and produce molecules high in potential energy. Exergonic means it gives off energy. Energy is a product. You start with molecules high in potential energy, release the energy, and your products are molecules low in potential energy. These two processes are inverses of each other. One stores energy, the other releases it. These are often confused with endocytosis and exocytosis. Remember that cytosis is referring to a cell. Look at this mathematically. If you want to mix 10 pounds of granola, you need to mix 2 pounds of nuts, 2 pounds of fruit, 6 pounds of grain. Total input equals total output. So look at the input and output here. 2 plus 2 plus 6 equal 10. Your reactants equal X for the molecules and Y for the energy. Your products must equal X plus Y. You must add Y amount of energy to the reactants to be able to make enough product. You can't expect to add a pound of nuts, a pound of fruit, and a pound of oats to get 10 pounds of granola. Make sure you understand the math. The same with an exergonic reaction. This time you're separating that granola back out. You start with 10 pounds of granola. Here's the nuts and fruit. You can calculate the energy, the oats, by how much is left to equal what you started with. Just to look at them side by side. Which one requires energy in? Which one puts energy out? Do you see how to figure the amount of energy needed or released? You won't have math problems with this, but you need to understand the basics. So what process is photosynthesis using? Starts with energy-poor reactants of carbon dioxide and water, but makes energy-rich ones. So it's endergonic. It had to add energy from sunlight to get products that are energy-rich out of those energy-poor ingredients. Look at cellular respiration, which is it. It starts with energy-rich molecules and produces smaller molecules with potential energy. It is exergonic. Energy coupling is showing the link between the processes. You will find that for each endergonic reaction, there is an exergonic reaction, like photosynthesis and cellular respiration. Doesn't it make sense that the energy that gets stored by one type of reaction will then be used by another? This is ATP, adenine, a 5-carbon sugar, ribose, RNA, and triphosphate. All the phosphate groups are negative. Remember, opposites attract. 3 negatives side by side are not happy. They're sitting there ready to pop off like releasing a spring. To release a phosphate group, the process is hydrolysis. Remember that this process was used to break down every single macromolecule? It's here too. Now, in the diagram, you've taken ATP, or adenosine triphosphate, and changed it to ADP, or adenosine diphosphate, prefix di means 2, plus a free phosphate group, and the bit of energy you released. What kind of reaction is this, endergonic or exergonic? Exergonic. Why? Releases energy. This energy is released in small controlled amounts that our cells use for different processes. So this exergonic reaction needs to have an opposite reaction to go with it. If this exergonic reaction involves breaking off a phosphate group, what do you think storing energy will involve? Yes, putting that phosphate back on. Hydrolysis, breaking apart using water. This is the process of transferring a phosphate group from an ATP molecule to a different molecule. What molecule might a phosphate group be attached to? How about a transport protein being used for active transport? This transfers energy to the molecule getting the phosphate group, or being phosphorylated. Energy given off by exergonic reactions can be used for phosphorylation. ADP is phosphorylated into ATP. It undergoes hydrolysis to release energy from endergonic reactions and forms ADP and free phosphate groups, which are then available to be phosphorylated again. Continuous energy coupling. Although the molecules are reused, the energy itself is moving one way. It's all eventually released as heat and must be replaced from outside the system, from our sun. These are different ways energy is used by our cells, different forms of work. Chemical work. Phosphorylation drives endergonic synthesis or building of product molecules. Mechanical work. Transfer of phosphate groups to special motor proteins in muscle cells causes the proteins to change shape and pull on actin filaments, those microfilaments, causing contraction. Some organelles use these actin filaments to assist their movement throughout the cell. Transport work. Phosphorylation of certain membrane proteins. Since it takes energy, this would be active transport. Our macromolecules have lots of potential energy. Why don't they just randomly break down and release all that energy? For a reaction to occur, an energy barrier must be achieved first. This barrier varies with the reaction. Think of a pack of matches. They all have the potential to burst into flame, but they don't. They need a boost from friction or some other form of heat first. Enzymes, as catalysts, can help. They don't help out with providing that activation energy. What they do is lower the amount of activation energy needed. The reactants are on the left with potential energy due to position. They're higher up. It takes a bit of extra energy to get over the energy of activation barrier represented by the wall here. There are ways to provide extra energy. Heat may increase the speed of a cellular reaction by increasing the amount of energy available so the energy of activation barrier can be overcome. But this comes with potential problems. But remember, many of the molecules in reactions are proteins. What can you do to proteins when you heat them too much? Denature. Then they won't work at all. Plus, you can't selectively heat just certain molecules. You've got to heat the whole organism. So instead, enzymes lower the activation energy needed to start a reaction. This allows the reactants to get started with less initial expenditure of energy and puts a control on the reaction. With a control, they won't be randomly having chemical reactions. Your macromolecules will not randomly break down. As I said, enzymes don't add any energy to the reaction, so they don't help get over the energy of activation barrier. They actually lower the barrier. And they do this without undergoing any permanent molecular change. What this means is that most enzymes can be used over and over again. If you want to make a peanut butter sandwich, you only need bread and peanut butter. But it would make it faster, easier, and nicer if you had a knife to spread the peanut butter. And you can use that knife again later, even after you've eaten your sandwich. Here's another way to illustrate it. You start with reactants. The black line represents the reaction without the enzyme added. It looks like a hump that must be gotten over, and that's what it is. You can see the energy of activation is still there with or without the enzyme added. The enzyme lowers it but doesn't remove it. The net change in energy is from the reactants to products and does not include the energy for activation. Enzymes are proteins. Of all the molecules, protein shape is most critical. Remember that shape determines function. You wouldn't use a screwdriver to drive that nail or a hammer to turn a screw. This is a reaction catalyzed by an enzyme. Start at 1. Here's your enzyme. This one is sucrase. Remember about enzymes ending in ASE? The specific shape on this side is called the active site. It's the specific shape that reacts with this substrate or the reactants that will be used in this reaction. The The substrate in this case is sucrose. The enzyme sucrase breaks down sucrose. When the substrate joins with the enzyme with a weak bond, the enzyme alters its own shape slightly to hold it. This is called induced fit. When it does this, it distorts the shape of the substrate, the sucrose. That weakens the bonds on it. Then it adds water. Why? What's happening? Hydrolysis. The bond in the polymer is now broken and the products are released. These products are glucose and fructose. Notice that although the enzyme temporarily changes shape, its molecular structure is unchanged by the reaction. It may help the reaction along, but it doesn't take part in it. Therefore, after the products are released, the enzyme is back in its original state and can accept a new substrate and do it all again. Not a new type of substrate, but a new molecule of the same substrate. Denaturing. Heat, pH change, salt concentration. I hope we've mentioned all this enough times that you're expecting questions on your exam. Some enzymes need helpers of their own. The ions would be inorganic molecules. The vitamins are organic molecules. If they're organic molecules, then they are also called coenzymes. Inhibitors sound bad, but that's not always true. In fact, the product of a reaction itself, if it's in sufficient abundance, may act as a feedback inhibitor and block production of more of itself. So how do these work, and why are they needed? In the top picture, you have the enzyme and its normal substrate, which fits nicely into the active site. In the bottom left, an inhibitor is shown. This one has part of the required shape and fits in the active site enough for it to stay there. Is this competitive or non-competitive? Yes, competitive. The inhibitor actually competes with the substrate for the active site. It's molecular musical chairs. In the bottom right, nothing has fit into the original active site, but something has fit into another site on the enzyme. And by doing so, the shape of the active site has changed, and the substrate can no longer fit. Which would this be? This is non-competitive inhibition. The inhibitor fits into the allosteric site instead of the active site. This causes the enzyme to change shape, including the shape of the active site. The substrate no longer fits, so the enzyme can't work. In both cases, when the inhibitor leaves, the enzyme is restored to full function. This is how a thermostat works. You set the preferred temperature. When it gets warm or cool enough, the temperature itself shuts off the heating or cooling. When the temperature shifts, they'll start back up. In feedback inhibition, a product molecule in a sequence is the inhibitor. It may be a direct product or one down the line a bit, as the illustration shows. Once the other product is used, the product inhibitor leaves the enzyme, allowing it to work again. So why are inhibitors needed? Enzymes need some controls, else they would be continuously breaking down molecules randomly. These guys work by interfering with enzyme function. Examples in your text are cyanide inhibits ATP formation during cellular respiration. Serine blocks acetylcholinesterase, an enzyme needed for nerve impulse transmissions. This causes paralysis of vital functions, not just muscle paralysis. Pesticides, such as malathion, also inhibit acetylcholinesterase, but the dosage is usually too low to affect us. It's why we have all the safe handling laws on some pesticides. While it takes a lot to affect an adult, children need a much lower dosage. Penicillin inhibits a bacteria cell enzyme needed for forming cell walls. This is an enzyme we don't have, as we don't have cell walls, so we don't have to worry about it affecting our own cells. Many of our antibiotics attack structures that bacterial cells have that we don't. There's lots more, but you can see how important enzymes are to us. We accumulate that knowledge. Then we use it to create these inhibitors that will do what we want. Technology in action. This is an example of a flow chart that's at the end of the chapter in the text. It's an excellent study tool. The answers are as follows. A, active transport. B, the concentration gradient. C, molecules such as water. D, facilitated diffusion. E, transport protein. What's the photo? Energy, though. Energy conversion, changing energy from one form to another.